All right, and continuing our discussion here with cell respiration, we're going to talk about how ATP is actually made. And the process uses the enzyme ATP synthase as well as the process of chemiosmosis is really what's going to ultimately generate the ATP. And if you recall, the inner part of the mitochondrial membrane has a lot of surface area, and there's lots of copies of the ATP synthase protein that's in there. And what this does is it uses the hydrogen ion gradient that we'll talk about here in a minute to turn, the kind of the complex of the protein to turn that, and that phosphorylates ADP into ATP. Okay, so what chemiosmosis is basically just a big fancy word that describes the movement of the hydrogen ions from a high concentration to a low concentration. Okay, so the mitochondrial membrane is going to generate this hydrogen ion concentration, and then as it flows from the matrix to the inner membrane space, you're going to get lots of energy that's in there. Okay, so what happens is you have this basically you have this bunch of cristae kind of on the inside, which is what I'm drawing for you here right now. And what you're going to do is you're going to pump the hydrogen ions across the membrane here. So we're going to have a high concentration of these hydrogen ions, as you'll see. And then as they flow back into the matrix, and that's kind of what this inside area here is called, is the matrix. Once they flow back into the matrix, that's going to take ADP plus phosphate, okay, and turn it into the ATP, okay? So that hydrogen ion gradient that exists, that's what's going to drive the synthesis of the ATP into that matrix of the mitochondria. So here's kind of what the ATP synthase molecule looks like, and here's this inner membrane space. So if you look right here, kind of go back, that's this spot right here. Okay, that's the intermembrane space. Okay, and then you can see that there's a high concentration of these these hydrogen ions right here. So that's what these things are. And as these things flow through the ATP synthase complex into the matrix, okay, so there's the matrix. So if I back up here, this spot that I colored in a little bit earlier here, this right here, this region in here, this is the matrix, okay? So as they flow into the matrix now, this is what's going to help take ADP plus the phosphate and turn it into the ATP. Okay, so this process of chemiosmosis, this is what's making use of this hydrogen ion gradient. It's called a proton motive force, and this is what's going to kind of be responsible for generating all of the ATP for us. Okay, so now if you think back to what we talked about very early on, the production of the NADH, which you see right here, and the FADH2, these are the electron carriers, and we get these from the citric acid cycle, we get these from glycolysis, and what they're doing is they're carrying the electrons, and when they drop them off at the electron transport chain, and this is on that inner mitochondrial membrane, okay, these are those cristae that you see, like for instance right here and here and here and so forth. These contain all of these proton pumps, which are going to pump these hydrogen ions across the membrane. Okay, so from this matrix, that's what this is right here, this yellow area, from the matrix into the inner membrane space, which is the orange area. And because they're charged ions, they can't get through the membrane. Okay, they won't be able to pass through because of this nonpolar region in here. They can only pass through the ATP synthase molecule. And when they do so, ADP and phosphate join together and give us the ATP. So we're going to make use of this gradient much the same way as water behind a dam. When you allow it to pass through the dam, you turn the turbine and you generate electricity. And this is this ATP synthase molecule right here. This is what's kind of acting as that turbine. And then the energy that we're harnessing from this concentration gradient that's being created, from the electrons that came from the potential energy in the bonds of the food, that's what's going to allow us to generate this ATP according to this process here. Okay, so we get about 36 to 38 ATP produced per molecule of glucose, and there's a few reasons why we can't really put an exact number on it. So the phosphorylation, the addition of the phosphate group to the ADP, at these redox reactions that we've been talking about, and these redox reactions are right here. So this is a reduced form, okay, and then what it does is it reduces this and this becomes oxidized, and then when the electron is transferred to the next carrier in the chain, this becomes reduced, and then this one becomes oxidized, then this is reduced and this is oxidized, and so forth. So these are the redox reactions, and they don't occur in an exact 1-to-1 ratio. And we have the NADH here. This is going to give us 3 ATPs, whereas the FADH2 is only going to give us 2. Okay, because it enters, if you look here, it enters lower in the electron transport chain. So if we look here, you can see higher up on the slide, this is where the NADH is entering. FADH2 enters a little bit lower, so therefore, the NADH allows us to pump many more hydrogen ions through as it passes all the way through this membrane here, whereas the FADH2 doesn't pump quite as many, and as a result, we get less ATP from the FADH2. Okay. The other thing is any of the NADH now that's being generated from glycolysis doesn't get in directly to the mitochondria. It has to be shuttled through a transporter. Okay, so if we look here at the transporter, here's glycolysis. This is in the cytoplasm. This NADH that's getting made here, this is what's that's getting made is getting funneled into the mitochondrion through a membrane shuttle. Okay, and it's just basically luck of the draw as to what it becomes when it gets shuffled in. Some of the electrons are transferred to other NADs, giving more NADH. In this case, we'd get 3 ATPs. Okay, whereas the luck of the draw may have it that the electrons get shuffled to an FADH molecule, and then this would result in the production of 2 ATPs. Okay, ATP. So what that would do, get it here, ATPs, what that would do is reduce the amount that we could ultimately make. Okay, so that's kind of what we're explaining with these words really right here, is that, it just basically, if NAD is the acceptor, we're going to get 3 ATPs. If FAD is the electron, then we're going to get 2 ATPs. So just minor differences, but it results in a difference in how much ATP actually gets made. And then some of this proton motive force is used to kind of uptake or to power the uptake of the pyruvate from the cytoplasm, and allow us to utilize that pyruvate as an energy source. So that brings down some of the proton motive force, which brings down some of the actual numbers of ATP that can actually be made. Okay. So, see what's going on here. There we go. Sorry. Okay, so you can see this is what I was talking about here. This is the uptake of pyruvate, okay, into the mitochondria, and that takes a little bit of energy, and that is what's responsible. That's the link reaction that we talked about in the last presentation, and that requires a little bit of energy. Okay, so if all of the proton motive force were used, we'd get a maximum of 34 ATPs here, plus the 4 that we get from substrate level phosphorylation, giving us 38 total. And again, the substrate level phosphorylation, a little bit of that is in glycolysis, and a little bit more of that is in the citric acid cycle. Okay. So that would be the maximum. And then the ATP that gets made in the mitochondria, and this has to get transported out and into the cell. So in other words, let me change my color here. ATP is getting made here, okay, in this matrix portion, the interior, but it's needed out here, okay, in the cytoplasm. So what we have is what's known as the ATP-ADP translocase molecule, okay, or enzyme, and it's a protein that's basically going to transport the ATP from where it's made in the matrix out into the inner membrane space. Okay, so in other words, what we're going to do is we're going to transport it right through here, okay, and it's going to be in this spot, and then it will very easily diffuse out into the cytoplasm where it's needed. And then the other thing that it does is, I mean, if you look at the name of it here, let's see, get going. I'm stuck a little bit again here. What happens is the ATP is getting shuffled out to where it's needed, but when it's burned, ATP becomes ADP plus phosphate. Well, that ADP plus phosphate gets shuffled back in on the other side of this membrane through that transport protein. And what that does is it allows us to use that to make more ATP. Okay, so that's how this thing is basically working. All right, so now let's see why seem to be a little bit stuck here. Okay, well, I'm going to stop this and I'll pick up with another part here in just a sec.